Welcome to the lecture on only the innate immunity. This is sort of a continuation of lecture 1 from immunology and serology because we talked about the immune system in general overall, and we talked about innate immunity and adaptive immunity. And so this lecture is just going to focus on innate immunity. Innate immunity includes both external defense systems and internal defense systems. And we're going to talk about what's involved in each one of these during this chapter, during this lecture. The innate immune system, these are the defenses against infection that immediately act when a host is attacked by a pathogen. It's composed of two different systems that work to promote phagocytosis. The external defense system includes anatomical barriers like skin, etc. We're going to go into those in more detail. The external defense system is designed to keep microorganisms from ever entering the body. The internal defense systems include cellular responses that recognize specific molecular components of pathogens after they have entered the system and gotten past our external defense system. The external defense system is composed of any physical, chemical, and biological barrier that work together to prevent infection from entering the body. This includes coughing, sneezing, reflexes to expel microbes, salivary glands, and this is tear ducts. These include an enzyme called lysozyme to break down any bacteria that enters or lands in the skin. Airways are coated with mucus and have cilia to help trap bacteria. The skin, it has an acidic pH in the sweat glands, and the sebaceous glands have fatty acids. And then in the stomach, there is gastric acid that lower the pH and create an acidic environment. So don't forget or keep in mind that lysozyme is an enzyme that's in our tears and saliva to help break down pathogens. The external defense system is composed of several different things, and we're going to talk about these individually. The skin makes a physical barrier, and these include secretions, skin secretions like with sweat glands or sebaceous glands that discourage pathogenic mycobacteria from growing. Now, there are some normal flora on the skin that also help with that, but right now we're just talking about the skin itself. The skin is composed of the epidermis and the dermis. The epidermis is very tightly packed epithelial cells coated in keratin, and the dermis is the connective tissue with blood vessels, hair follicles, and our sebaceous and sweat glands, and a few white blood cells. Lactic acid and fatty acids on the skin maintain the pH at approximately 5.6, which is acidic. And then psoriasin is a small protein produced by skin cells that has antibacterial effects. Isn't that interesting? The respiratory tract is coated with mucus secretions, and it also has cilia, which are moving finger-like projections that block bacteria from adhering or attaching to epithelial cells. Also, the movement of coughing and sneezing moves pathogens out of our respiratory tract. Urine, both the acidity of the urine and the outward flow of urine are part of the external defense system that help remove potential pathogens from the genitourinary tract. The digestive system is made up of, as far as external defense system is concerned, the stomach includes hydrochloric acid, which keeps the pH of the stomach very low as 1, prohibiting microorganisms growing. Also, the digestive tract has normal flora or microbiota to help keep pathogens from establishing themselves. So the normal flora are already growing there, so their growth helps keep pathogenic bacteria or viruses or parasites from establishing themselves. So it's very important. The internal defense system consists of pathogen recognition receptors, which are on different kinds of cells, and we're going to talk about that next. Acute phase reactants, we're also going to define that. Inflammation, phagocytosis, and natural killer cells. Those are part of the internal defense system. Pathogen recognition receptors. These are on molecules, and they recognize molecules unique to infectious organisms. Pathogen recognition receptors are found on macrophages, dendritic cells, neutrophils, eosinophils, monocytes, mast cells, T cells, and epithelial cells. Acute phase reactants are soluble factors found in serum. These factors increase rapidly within 24 to 48 hours in response to infection, injury, or tissue trauma. Acute phase reactants facilitate contact between microbes and phagocytic cells, so they facilitate contact between, say, bacteria and our monocytes. They also mop up and recycle important proteins after phagocytosis. And this is important because we're going to learn later on that inflammation causes a lot of chemicals to be released in our body. You may have heard in the news in relation to coronavirus, the cytokine storm. These are chemicals inside your body that are released all over, and it's kind of like mini explosions all over your body, and it can do tissue damage. So acute phase reactants also mop up and recycle these important proteins after phagocytosis to limit the destruction of the host. Acute phase reactants, it's also important to remember that these are non-specific indicators of inflammation. So I'm going to show you a list of an example of the most commonly discussed acute phase reactants. And these cannot be used to diagnose any specific disease. It's just used as a non-specific indicator of inflammation, as in, yeah, there's some kind of immune response going on. So the acute phase reactants of particular importance include C-reactive protein, and its function is to serve as an opsonization component and also in complement activation. And complement has its own chapter. Serum amyloid A activates monocytes and macrophages. Alpha-1 antitrypsin is a protease inhibitor. That one's not so important for us. Fibrinogen is involved in clot formation. So keep this in mind, especially with COVID-19. One of the things that's happened in patients was microclots all over the body. And think about it. This fibrinogen is an acute phase reactant, and it causes clots. So if you're in any kind of infectious cycle, these acute phase reactants are going to be elevated. Fibrinogen is one of those. So you would expect microclot formation. Haptoglobin is a protein that binds hemoglobin. That's its normal job. It's also an acute phase reactant, so it would be elevated in inflammation or infection. Ceruloplasmin is a protein that binds copper and oxidizes iron. And complement C3, that's one component of complement. C3 opsonizes and lyses. That's its function. So next we're going to talk about specifically the process of inflammation. And I've put my favorite inflammation picture, the hangnail. Everybody's had one. You know what it feels like and what it looks like. So inflammation is the body's overall reaction to injury or invasion by any infectious agent. The cardinal signs and symptoms of inflammation are these four, and I put the Latin term beside the commonly used term for the cardinal signs and symptoms: redness, swelling, heat, and pain. So redness is caused by the increased capillary permeability and red cells that rush to the area. Swelling is caused by blood and white blood cells flying to the area and swelling the area. Heat is caused by the fever that's induced, because think about it, I'm going to show you a picture on the next slide, And we talk about there's all these chemical reactions going on during inflammation, these acute phase reactants. And so you've probably learned in chemistry that one of the signs of a chemical reaction or indicators is heat is given off. So that's what's going on. And then pain, because hangnails are so painful. And the Latin terms are rubor, tumor, calor, and dolor, just for fun. So here's a picture of inflammation if you get a splinter. So you get a splinter, the splinter punctures the skin. Damaged tissues released histamines that increase blood flow to the area. So you have capillary permeability. Histamines in the granules of our phagocytes release phagocytes and clotting factors to the wound. Then phagocytes engulf the bacteria, the dead cells, and cellular debris, and then platelets move out of the capillary to seal the wound. So all that is going on when you get just a little splinter or hangnail. It's very interesting. So to recap what we learned in Lecture 1, how do those white blood cells get to the tissues? Through diapedesis. The neutrophils use their pseudopods and squeeze through the gaps in the epithelium. And another way that we can measure inflammation, we can measure those acute phase reactants in our blood, but we can also do this test called erythrocyte sedimentation rate. So we put blood in whole blood, we put it in these long columns with measurement, with millimeter markings on the outside, and we let it sit for 1 hour. And we measure the distance that the red cells fell within an hour. And so if you have an increased erythrocyte sedimentation rate, so if these all look normal. So if you had yellow all the way down to here and the red cells were just down here, that means that the red cells fell all this way in an hour. And that is how we measure inflammation in a very non-specific way. Okay, next we talked about neutrophils in the first lecture as well. Neutrophils are the primary cell involved in the acute inflammatory response. Neutrophil immigration may last 24 to 48 hours, and it is proportional to the level of chemotactic factors present in the area. So remember, chemotaxins are chemicals that draw phagocytes to an area. Migration of macrophages from surrounding tissues from blood monocytes. Remember, monocytes, or we call monocytes in the peripheral blood monocytes, and monocytes in the tissue are called macrophages. The migration of macrophages to the tissues peaks at 16 to 48 hours. Macrophages attempt to clear the involved area through phagocytosis, and in most cases, healing is completed pretty quickly. But in cases of chronic inflammation, tissue damage can occur because remember, with inflammation, you've got all those chemicals. You've got the pressure of the swelling, the increased blood flow that puts pressure on tissues. Plus you have those chemicals that can cause tissue damage and eventually loss of function. And then of those acute phase reactants that were in our table, CRP, C-reactive protein, is the most widely monitored of the acute phase reactants, and it's the best blood test indicator of acute inflammation. Next, we're going to move on to phagocytosis. And the cells most involved in phagocytosis are neutrophils, monocytes, macrophages, and dendritic cells. Those contain an NADPH oxidase enzyme that generates superoxide. And you probably remember from a general biology course that NADPH is part of the energy generating cycle. The oxygen species like hydrogen peroxide that are derived from superoxide together with enzymes like our lysozyme and proteases liberated from the granules are what is used to kill ingested microbes. So here are the steps in phagocytosis. The first one is adherence. That means there's physical contact between the phagocytic cell and the microorganism. Then engulfment, where there's an outflowing of cytoplasm to surround the microorganism. Formation of the phagosome, where the microorganism is completely surrounded by part of the cell membrane. And then there's granule contact. So lysosomal granules contact and fuse with the phagosomes. Then there's the formation of the phagolysosome, where the contents of the lysosome, and remember, these are powerful chemicals, those are emptied into this membrane-bound space, and digestion of the microorganism by hydrolytic enzymes occur. Then the last stage is excretion. So the contents of that phagolysosome are expelled to the outside by exocytosis. So during phagocytosis, this is a highly combustible reaction. We have formation of hydrogen peroxide and the hydroxyl radical. Hydrogen peroxide and hydroxyl radical. Those are very important chemicals for the oxidative burst. So now I'm going to take you to YouTube. This is such a cute video. This is a video of an oxidative burst in a macrophage. It ingests the bacteria and it gets really bright. That was it. We're going to watch that again. It goes so fast. Okay, so the phagocyte is going to engulf the particle, and then there's the energy burst. Wow. And that was exactly what's going on inside a macrophage during phagocytosis. Phagocytosis, remember from lecture 1 also that opsonization enhances phagocytes, and opsonins are serum proteins, like some of our acute phase reactants, that attach to a foreign substance and help prepare it for phagocytosis. Because remember, our opsonins are kind of like salt and pepper. And in the Greek terminology, opsonin means preparing to eat. So examples of opsonins include C-reactive protein, complement components, and antibodies. Natural killer cells are our first line of defense against cells that are virally infected, also against cells that are infected with other intracellular pathogens and tumor cells. Natural killer cells recognize any damaged cell or any cell that lacks MHC class I markers. Because remember, MHC class I markers are the ones that tell natural killer cells that those cells belong here, do not kill them. So these natural killer cells can eliminate cells without prior exposure, giving the immune system time to activate the adaptive response of specific T and B cells. And that's going to be the whole topic of our next lecture. Natural killer cells are stimulated by exposure to cytokines, and when they are activated, they produce additional cytokines. Hello, the cytokine storm. And natural killer cells influence innate and adaptive immunity. So let's summarize what we discussed in this lecture. The innate immunity encompasses the body's normally present defense mechanisms, like skin. Innate immunity is characterized by lack of specificity, no need for prior exposure, and a similar response with each exposure. Our external defenses are structural barriers and secretions that keep microorganisms from ever entering the body. Our internal defenses include cells capable of phagocytosis, like acute phase reactants that enhance phagocytosis. Pathogen recognition receptors are molecules on host cells that recognize substances found only on pathogens. And once receptors bind a pathogen, phagocytosis can take place. Acute phase reactants are serum proteins that increase rapidly within 24 to 48 hours in response to infection or injury. Inflammation is the body's response to injury or innate invasion by a pathogen. Natural killer cells kill target cells that are infected with a virus or other intracellular pathogen, or those cells that don't have an MHC class I marker. And natural killer cells are a link between the innate and adaptive immune systems. And the adaptive immune system is up next.